Thank you very much for inviting me to speak today about the microenvironment in myelodysplasic syndromes. Thank you. I do not have any conflict of interest to declare, and I would like to thank to the Fondation Les Amis de Rémy qui nous soutient. I would like to thank to the Foundation Les Amis de Rémy, which support our research activity in a hematological laboratory from University Hospital of Saint-Étienne. I will start with a celebration. In 2014, we celebrate 100 years from the seminal book on the origin of malignant tumors, which was written by German zoologist Theodor Boveri, and that stated two claims concerning the cell biology with implication in cancerology. The first one is the proliferation. The first one is the proliferation is the default state of cells, and the second, the cancer is a cell-based disease. Shortly after Boveri's death, Whiteman have introduced another idea that cancer is a cell-based disease. It means a cancer cell is a somatic mutated cell. So this was the cornerstone for the somatic mutation theory. And nowadays we know that cancer is a collection of over 200 diseases with have been very well described, starting from somatic mutation theory in Hallmark of Cancers by Hanahan and Weinberg. But what cannot explain the somatic mutation theory? After 100 years of research focused on somatic mutation theory, only a third of risk of the cancer is due to the environmental factors and to the inherited predisposition. For the majority, we know is bad luck. So isn't it time to switch to the tissue organization field theory? But to talk about tissue of Toft theory, we have to introduce the main actors which are involved in bad soil or microenvironment. For hematopoiesis, the microenvironment is provided by bone marrow and in the so-called hematopoietic niche. Two very important actors in this hematopoietic niche are the mesenchymal stromal cells and the hematopoietic stem cells. Concerning the mesenchymal stromal cells, we have the most immature and multipotent mesenchymal stromal cells, which reside in the fraction 45 negative, lineage negative, 271 positive, and PDGFR receptor beta low or negative expressor. Moreover, these cells may be divided by their expression for MCAM molecules into two subpopulations, and those which are CD146 positive, it seems to be those which have the most supportive capacity for the hematopoietic stem cells. Also, the more mature osteoprogenitors may regulate the hematopoiesis differentiation and proliferation through synthesis and secretion of microRNAs. The most important interactions which are between mesenchymal stromal cells and cancer cells are two types. Indirect interactions mediated by soluble factors that might transfer biological information between neighboring cells, but most important are the direct interactions. And for hematopoiesis, the most important is mutual activation of notch signaling. If we go back to myelodysplasic syndromes, the first evidence which bring incrimination of myelodysplasic syndromes in MDS pathogenesis has been proved by Riemakers in 2010, and he showed that DICER1 deletion in osteoblast progenitors, this is a gene involved in microRNA processing, could lead to MDS in mice model. Also, SBDS deletion in CEBPA mutated HSPC could lead neutropenia, but no MDS or acute myeloid leukemia. However, when we see this SBDS deleted and CEBPA mutated HSPC in a mice carrying SBDS deletion in osteoprogenitor cells, this could lead myelodysplasic changes. The mechanism underlying of this abnormal hematopoiesis driven by deletion of SBDS is inflammation, because deletion of SBDS in osteoprogenitor cells induce activation of P53 signaling pathway, and this will lead to synthesis and secretion of alarmins, S100A8, A9. And these alarmins act on toll-like receptor 4 and induce HSPC apoptosis. Also, the accumulation of alarmins in microenvironment induce oxidative stress and genomic stress on hematopoietic precursor cells. In addition, alarmins act on a group of cells which are immune regulatory cells named myeloid-derived suppressor cells, and they link to molecules CD33 and toll-like receptor 4 and drive activation of NF-κB with synthesis of pro-inflammatory cytokines and inflammasome assembly. Another evidence is brought by Code in 2014 and constitutively activate mutation in beta-catenin in osteoprogenitor cells, induce accumulation of beta-catenin in nucleus, and overexpression of a notch ligand named JAGID1 on osteoprogenitors that may thereafter induce of notch signalization in hematopoietic precursor cells with impairment of myeloid and lymphoid differentiation of hematopoietic precursor, and that will allow the extramedullary hematopoiesis also. But this fetal osteoblast with activated mutation for beta-catenin, when we seeded in the ex vivo system in the presence of a cocktail of cytokines, appropriate cocktail of cytokines and growth factor, this may recover the differentiation capacity of hematopoietic precursors, and also it has been shown that they are not able to transfer acute mild leukemia in offspring. In another relevant article showed that engraftment of CD34 selected cells from MDS patients in an NSG mice is not able to induce myelodysplasic syndromes. However, if we co-transplanted, if the authors decide to make co-transplantation of CD34 selected cells with mesenchymal stromal cells from myelodysplasic patients, this lead a very good engraftment. And in this series of experiments, they show that the remarkable efficiency of engraftment of co-transplanted cells after 10 weeks post-transplant. Also, they show that the capacity of myeloid cells of the hematopoietic precursor is skewed to myeloid output in both situations if we if we put together CD34 positive cells with myelodysplasic mesenchymal stromal cells. The RNA screening showed in myelodysplasic mesenchymal stromal cells showed that the most different expressed genes are those related to inflammation and adhesion molecule process, related processes, and with stromal stimulation. Also, exposure of healthy mesenchymal stromal cells to MDS mesenchymal stromal cells, this could lead to altered gene expression, as showed here by high expression of leukemic inhibitory factor. In our experience, the mesenchymal stromal cells selected based on two specific markers, STRO1 and CD73, are different capacity of proliferation and different functionality in primary cultures. For example, the mesenchymal stromal cells from high-risk MDS, they have a cluster of aberrant proliferation compared with healthy mesenchymal stromal cells selected from age-matched donors. And in low-risk MDS cases, we have accumulation of large, thin, and and large, thin, and abnormal cells that features which may be related with altered actin organization. Also, the cells from mesenchymal stromal cells, which were selected from low-risk MDS, they show a low capacity of proliferation, and as is shown here, and also in both situations, low-risk and high-risk MDS, the mesenchymal stromal cells have a lower clonogenic capacity. The most recent data show that selected stromal cells from low-risk MDS, they have a higher propensity to develop an adipogenic differentiation in detriment of osteoblastic differentiation. So, which will be the mechanism? In the normal situation, in the normal bone marrow, the TGF-beta have a negative inhibit adipocyte differentiation. Instead, in myelodysplasic syndromes, the high levels of TGF-beta trigger a signalization through SMAD2/3 that promote adipocyte differentiation through releasing of microRNA 21, and these promote fibrosis and anemia, APO-resistant anemia. It was shown that inhibiting TGF-beta, this anemia can be recovered by recovering the late-stage erythropoiesis. Mesenchymal stromal cell expansion is an adhesion-dependent process. First line show you that clonogenic capacity of mesenchymal stromal cells is highly correlated with their expression for CD44 adhesion molecule. Also, the proliferation is an adhesion-dependent process because he is highly correlated with alpha-5 integrin expression. But also the impairment of growth kinetics of mesenchymal stromal cells in myelodysplasic syndromes might be correlated with a derepression of cyclin inhibitor, CDKN1A, and also in low-risk MDS may be correlated with high senescence. We want thereafter to know what's happened in downstream of integrin stimulation, and the most important molecule which govern a lot of cellular processes as survival, cell proliferation, invasion, and migration is the molecule named focal adhesion kinase. We know that this molecule is correlated with differentiation capacity of mesenchymal stromal cells, and also with interactions with endothelial cells and lymphocytes in bone marrow. And we have so that in high-risk MDS, expression of FAC is upregulated, also its activation, and this was correlated with high level of expression for paxillin and HSP90. And recently we have observed that graduate increase of expression for FAC is correlated with disease advancement toward acute myeloid leukemia. The colocalization analysis showed a high superposition of these three proteins, paxillin, HSP, and PFAC, in nuclear region, and this was correlated with high proliferative capacity of mesenchymal stromal cells selected from high-risk MDS, which is a mechanism we thought that HSP90 may activate by phosphorylation FAC, and thereafter FAC will activate and phosphorylate paxillin and will modify its conformation with arrest of paxillin in nuclear region, and this might promote cellular proliferation by two mechanisms. By inhibition of a suppressor of a tumor suppressor, H19, and by induction of DNA synthesis. So MDS is the best model to explain the heterotypic reciprocal signaling disease because early epigenetic events which act on normal hematopoietic stem cells induce their modification with initiation of MDS disease initiating cell. And these cells have capacity to reprogram mesenchymal stem cells to proliferate and to inhibit the normal compartment of mesenchymal stromal cells, and to allow development of a mutational field which allow genetical evolution of toward acute myeloid leukemia. We believe that if our hypothesis is right, we believe that inhibitor of FACs, we can solve the problem with mesenchymal stromal cell compartment, but also with hematopoietic compartment, because a high expression of FAC in mesenchymal stromal cells in high-risk MDS was associated with low clonogenic capacity in hematopoietic compartment. Also, maybe it's a process related to an abnormally expressed SDF1 in stromal mesenchymal cells selected from low-risk MDS. And to conclude, we show in the I am showing in these few minutes that we can induce MDS-like malignant transformation in mice, but in the presence of mutated abnormal mesenchymal stromal cells, and hematopoietic stromal precursor cells might induce abnormal activation and focal adhesion kinase in mesenchymal stromal cells. And these mesenchymal stromal cells, which overexpress FAC, may contribute to tumor progression and protection. So I would like to thank you for your attention and to present my colleagues, which are working in research in our lab. And I would like to thank especially to Alexa, which is a PhD student involved in research in microenvironment in myelodysplastic syndromes. And if in a few years you will welcome in your office an MDS patient, maybe you will be able to say that We manage this because we developed a new molecule which target also hematopoietic precursor cells and microenvironment mesenchymal stromal cells. Thank you for your attention.